So I want you to try something for me right now. Just take a look around wherever you happen to be and find something with a really vibrant color.
Like a bright red coffee mug on your desk or, maybe the green leaves of a houseplant.
Yeah, exactly. Or just a patch of blue sky out the window. Just focus on it for a second, because usually when we talk about a biological process, like seeing that specific color, there's this expectation of a simple, almost mechanical explanation.
Right. We tend to think of the eye like a digital camera. Light comes in, hits a sensor, the sensor fires a wire to the brain, and the brain just says, there it is, red.
It feels clean. And I think it's comforting. We like things to be straightforward, but the reality is so much weirder.
Oh, it really is. I mean, once you step into the world of molecular biology and atomic structures, that simple camera metaphor just completely falls apart.
It really does.
We're suddenly looking at this microscopic landscape that is dizzyingly complex. It's an intricate dance of atoms constantly shifting and balancing.
It is the absolute definition of molecular gymnastics. And, well, that is exactly what we are unpacking today. We're looking at a truly groundbreaking 2026 science paper by Peng and colleagues.
Right, the one titled Cryo-Electron Microscopy Structures of Human Cone Visual Pigments.
Yes, that's the one. And what's so fascinating about this specific research is that for decades, we've known about the cells in our eyes that let us see color.
Yeah, we've known their basic function for a long time.
Right, but we have never actually seen their exact three-dimensional molecular blueprints in action. Not until this paper.
Exactly. The researchers used a technique called cryo-EM. That's where you flash freeze these proteins mid-action and take thousands of electron pictures to build a high-resolution 3D map.
Which is just mind-blowing. So our mission for this deep dive today is to pull back the curtain on those maps. We're going to explore the tiny atomic differences that allow you to see distinct colors.
And why our daytime color vision has to operate at such lightning speed.
Yeah, and what exactly happens down to the structural level when that color vision breaks down.
And to really grasp any of that, we have to start at the foundation, right? Before we can understand how we see different colors, we need to understand how we catch light in the first place.
The hardware of vision.
Precisely. If we look at the bigger picture, your retina, that's the sensory screen lining the back of your eye, it has a strict division of labor. You have rod cells, which are basically your night vision goggles.
Right. They are optimized for dim light using a protein called rhodopsin.
Exactly, peaking at around 500 nanometers. But rods don't see color. To see that red coffee mug, you rely on a completely different set of cells called cones, which operate primarily in daylight.
And as humans, we are trichromatic, right? We have 3 distinct types of cone proteins, or opsins, handling different parts of the spectrum.
Yep, we have the long-wavelength opsin, or LWS, for red light around 560 nanometers. Then the middle wavelength opsin, MWS, for green at 530 nanometers.
And the short wavelength opsin, SWS, for blue down at 420 nanometers. But regardless of whether it's a rod or a cone, there is a hidden MVP inside all of them.
Oh, absolutely.
It's a tiny molecule derived from vitamin A called retinal.
That's right. Retinal is the actual light catcher. Now, to picture this, you have to imagine the opsin proteins as these tiny structural barrels embedded in the membrane of your eye cell.
Okay, barrels.
Yeah, and they are made up of 7 tiny pillars, or transmembrane helices, crossing the membrane. Hidden deep inside the center of that barrel is the retinal molecule.
And it doesn't just float freely, does it?
No, it is chemically tethered to one of those pillars by a highly specific nitrogen-carbon double bond.
A shift-base linkage.
Exactly. It holds the retinal perfectly in place like a dog on a very short, very strong leash, usually at a specific lysine amino acid like K312 or K293.
Got it.
So in the dark, the retinal molecule is bent. It has a physical kink in its structure called 11-cis retinol.
But the magic happens when a single photon of light hits it.
Yes. In fractions of a picosecond, that bent molecule absorbs the light energy, breaks its internal bonds just long enough to rotate, and forcefully snaps it to a perfectly straight line, becoming all transretinal.
I absolutely love visualizing this mechanism. Imagine retinal like a spring-loaded mousetrap hidden inside that protein barrel. The photon of light is the mouse that trips the wire.
Snap.
Snap. The molecule violently straightens out. And because it is physically tethered to the inside of that barrel, when it snaps straight, it acts like a lever, right? It physically shoves the surrounding protein pillars outward.
It's a massive movement on an atomic scale. One of the main pillars swings outward, and that outward swing changes the shape of the bottom of the barrel, creating a new opening.
It forms a docking station.
Right. A messenger protein inside the cell, specifically a G protein in this study, recognizes that new shape, binds to the docking station, and boom, the electrical signal is fired off to your brain.
Okay, so before we move on, let's just do a quick recap for you, the listener, to make sure we're all on the same page. Light hits the retinal molecule.
Retinal changes shape from bent to straight.
The protein barrel is forced to open up.
And that allows the G protein to dock, and the signal is sent.
Perfect. Which naturally leads me to a massive question. If this core mousetrap mechanism is exactly the same for all vision, if a photon just makes a molecule snap straight, how on earth does the protein know whether the light hitting it is red, green, or blue?
That is the perfect question, and it takes us right into the heart of what this cryo-EM paper uncovered regarding the red and green opsins. It comes down to a concept called electrostatics, and essentially how you babysit a volatile chemical reaction.
Okay, babysitting. Walk me through this.
Well, remember that chemical tether holding the retinal molecule in place?
The shift-base linkage, yeah.
Right. Well, it carries a positive charge. It's protonated. In the microscopic world of molecular physics, a strong positive charge sitting all by itself is highly unstable. It wants to react with something.
Oh, okay.
So, to keep the whole system stable while it waits for light, it needs a molecular babysitter. It needs a negatively charged molecule positioned right next to it to balance it out, which biologists call a counterion.
Opposites attract, they cancel each other out, and things stay calm.
Precisely. Now, in your dim light rod cells, they handle this very cleanly. They have a specific negatively charged amino acid, E113, acting as the babysitter in the dark.
And then when the light hits?
When the rod absorbs light and activates, the protein shifts, and a different negative amino acid, E181, tags in to take over babysitting duties. It's a highly controlled, very stable handoff.
What the researchers found in this paper is that the red and green cone opsins do something totally fundamentally weird.
Yeah, they really do.
This blew my mind when I was reading through the notes. In the dark, the red and green opsins don't just use an internal amino acid. They actually reach out into the surrounding cellular fluid, grab a free-floating chloride ion at position H197.
And physically wedge it into the protein structure to help babysit that positive charge alongside E129.
Right. But then when the light hits and the protein activates, they just drop the chloride entirely. They ditch it.
They ditch it, and instead they rely on a completely different pillar of the protein to provide the negative charge. They use E102 on transmembrane helix 2.
It is a massive divergence in structural engineering. I mean, they're using an entirely different part of the protein architecture to stabilize the active state compared to rods.
Furthermore, the paper notes that the red and green cones completely lack a specific triad of amino acids that rods use.
The E122, H211, W126 triad, right?
Yes. Rods have this 3-part molecular vice grip that holds the activated, straightened retinal incredibly tightly. The cones just don't have it.
Wait, I have to stop you there because it makes no sense to me intuitively. If the rod cells already evolved this perfectly stable, highly controlled babysitting system with a tight vice grip, why would evolution completely reinvent the wheel for daytime color vision? Why start dropping chloride ions and using a looser, weaker grip?
It comes down to one absolute necessity, and that's speed. You have to remember what these cells are designed to do. Rods are built to see a single photon of light in the pitch dark. To make sure your brain actually registers that tiny signal, the rod cells need to hold on to it for a relatively long time, about 100 milliseconds. That's why they need that tight stabilization triad. They clamp down and hold the signal.
But cones operate in bright daylight, where you are being bombarded by trillions of photons constantly.
Exactly. If your cone cells held on to every single light signal for 100 milliseconds, your daytime vision would be a completely blown out, overexposed blur.
It would be like taking a long exposure photograph of moving traffic. You wouldn't see the individual cars, just massive, blinding streaks of light.
That's a perfect analogy. You need the cell to fire and then immediately reset so it can catch the next photon. Because the red and green cones lack that tight rod stabilization, and because they use this weird alternative E102 babysitting network that drops the chloride, their active state is structurally much less stable.
They literally cannot hold a pose.
Right. They collapse and decay back to their dark, resting state in just 3 to 10 milliseconds.
Wow. That is at least 10 times faster than the night vision rods.
Yes. That highly engineered instability is the structural reason you can watch a fast-moving baseball in broad daylight perfectly clearly. But if you try to track a fast-moving object in the dim moonlight, It looks like a smeared ghost.
So the instability is a feature, not a bug. Night vision is this tight, slow vice grip to ensure the brain gets the message, but our day vision is this loose, fast resetting system so we can process high-speed reality.
Exactly.
Okay, time for another quick summary for you. Red and green opsins use a unique E102 counterion system, and they drop the chloride when activated.
And because they lack those rod-like stabilizers, they can reset incredibly fast.
Brilliant, but thinking about this, if red and green vision require the specific, looser architecture to work fast, how does the blue opsin fit into this? Blue light is fundamentally different, right?
It is. Blue light has significantly higher energy and a shorter wavelength than red or green light. And if the red and green opsins are closely related siblings that share a lot of this architectural philosophy, the blue opsin is the eccentric, strange cousin that breaks all the rules.
Well, the researchers managed to map the short-wavelength-sensitive, or SWS opsin, the blue one, at an incredible 2.61 angstrom resolution.
Which is just astonishing.
Right. I mean, how small is an angstrom, really?
Oh, it's vanishingly small. 1 angstrom is 1/10,000,000th of a meter. So at 2.61 angstroms, you aren't just seeing the general shape of the protein, you're seeing individual atoms.
And they found some wild structural anomalies that tune this thing to catch high-energy blue light.
Yeah, to catch blue light at 420 nanometers, you can't just use the standard mousetrap. You have to dramatically shift how much energy it takes to trigger the retinal molecule. The paper reveals that the blue opsin accomplishes this by building a highly polar microenvironment directly surrounding the tethered retinal.
Okay, let's break down polar environment, because that sounds like a chemistry textbook.
Fair enough. Instead of typical neutral amino acids, the blue opsin packs the space around the retinal with what they call a serine ring.
Right. 4 specific polar serine amino acids.
Yeah.
S87, S183, S289, and S292.
Exactly. And polar molecules have strong partial positive and negative charges. They're like tiny magnets. By packing them closely together, it creates an intense electromagnetic field around the retinal molecule.
Right, and I like to think of this electromagnetic field like tuning a guitar string. If you want a string to play a higher pitch, you tighten the tuning peg, increasing the tension.
I love that.
The polar ring in the blue opsin essentially tightens the molecular tension on the retinal. Because of that intense surrounding field, it requires a much higher energy pluck, a high energy blue photon, to overcome the tension, make the molecule resonate, and snap it into the straight position.
That's a fantastic way to picture it. The polar field changes the activation energy, but that is just the spectral tuning. Structurally, the way the blue opsin handles the physical force of that snap looks like a bizarre building code violation.
It really does. Let's talk about the bungee cords.
Right, so the first major structural anomaly is an extra disulfide bond, specifically between C84 and C296.
And a disulfide bond is a very strong, permanent covalent link between two parts of a protein, right?
Yeah. None of the other visual opsins, not the rods, not the red or green cones, have this specific bond. It physically ties two of the moving protein pillars directly together, severely restricting how much they can flex.
So imagine building a house, but you decide to strap the roof down to the load-bearing walls with heavy-duty steel bungee cords. That's this extra bond. It makes the top of the structure incredibly rigid. But then the biological architects do something seemingly insane at the bottom of the structure.
They remove the foundation.
Right.
In almost every other protein of this type, there is a bulky amino acid called D2.50 that acts as a universal anchor point at the bottom of the barrel. It holds a tiny, stable network of water and sodium that keeps the whole protein grounded when it's inactive.
But in the blue opsin, they swapped out that massive anchor molecule for glycine, G80, which is the absolute smallest amino acid available.
Yes. By swapping a bulky anchor for a tiny placeholder, a massive cavity opens up inside the base of the protein. It's a structural void. This missing anchor allows a continuous, chaotic channel of water molecules to rush all the way up into the core of the protein.
So just picture this biological machine for a second. You have removed the foundational anchor, flooding the basement with water, but you've tied the roof together with steel bungee cords. That sounds like a terrifyingly precarious setup. It feels like it should just fall apart.
It is intensely precarious, but again, you have to look at the functional payoff. By combining the rigid disulfide bond at the top with the missing foundation anchor at the bottom, the blue opsin's active state has a notoriously short lifespan, even faster than the red and green opsins.
It's built to self-destruct, or at least collapse back to baseline, almost instantly.
Precisely. Upon activation, the immense structural tension pulls it back into its dark state so rapidly that it gives us razor-sharp temporal resolution for blue light. It completely avoids any lingering ghost signals that could muddy our vision in bright, high-energy light environments.
Form follows function down to the literal atomic level.
It really does.
Okay, quick recap time. Blue opsins use a cooling serine ring to tune to blue light. And they have this weird combination of a missing anchor and a bungee cord bond to achieve hyperfast resets.
Nailed it.
We were looking at absolute microscopic perfection here. A delicate, hyperfast balance of chemical springs, specialized babysitters, magnetic tuning fields, and bungee cords. Which brings us to a somewhat darker question. What happens when our DNA makes a typo in the blueprint of this intricate machine?
And that leads us to the clinical implications of this research. What happens when the canvas tears? We're talking about color vision deficiencies.
Now, a lot of standard color blindness issues happen because a gene is just missing. Like the red or green gene gets deleted during cell division because they sit so close together on the X chromosome.
Right, that's fairly common.
But the researchers in this paper mapped out something much rarer and honestly much more damaging: missense mutations. This is where you have the full blueprint. But just one single amino acid out of hundreds is swapped for the wrong one during the manufacturing process.
Let's look at one of the cases they analyzed. There's a specific mutation called P187S. That's where a proline is swapped for a serine.
And proline naturally has a sharp, rigid corner in its chemical structure, right?
Yeah, exactly. So it's swapped for a molecule that is straight and flexible.
It's like replacing a steel elbow joint in a pipe with a straight piece of rubber tubing.
Precisely. Because that sharp corner is gone, the protein chain literally cannot make the necessary turn as it's being built. The structural solding just fails, and the entire protein collapses on the cellular assembly line.
Oh, wow.
In another case they looked at, the C203R mutation in the red and green opsins, it destroys a crucial structural bond with C126. Basically, it breaks those structural bungee cords we just talked about.
And without that bond holding the pillars together, the protein loses its integrity.
Right, it falls apart, leading to a severe condition called blue cone monochromacy.
Wait, I need to pause here because this completely changes my understanding. When someone has this specific type of color blindness, these single atom typos, it's not just that their biological sunglasses are tinted the wrong color.
No, not at all.
The entire protein machine is actually structurally collapsing in the eye.
Yes. And that is a critical clinical distinction to make. It's not just a benign filter change where everything looks a little washed out. When proteins misfold, they don't just disappear. They get stuck inside the cell's manufacturing center.
Yikes.
If enough of these mangled, collapsed proteins build up, it causes massive cellular stress. This can actually trigger programmed cell death. So a mutation that you might initially think just causes a color vision deficiency can actually turn into a progressive degenerative disease. The cone cells themselves literally die off under the stress of the broken machinery.
Wow. A single typo causing a complete cellular meltdown. But the paper also showed that it's not always lethal to the cell, right? You can have typos that just change the tint without destroying the whole machine.
Absolutely, and this gets into spectral tuning. The researchers modeled a mutation in the green opsin, changing an alanine to a threonine at position 285.
So a relatively small molecule is swapped for one that is just slightly bulkier.
Right. It doesn't break the protein, but just adding that tiny bit of extra molecular bulk physically nudges the retinal and shifts its light absorption by about 12 nanometers. It literally redshifts the color the cell can see, moving its sensitivity from pure green at 530 nanometers slightly toward yellow-red at 542 nanometers, all without destroying the structural integrity.
Just swapping one atom for a slightly larger one moves your entire reality down the rainbow.
It really highlights the immense fragility of our perception. Our entire daily experience of a rich, colorful world relies on these exact, delicate atomic sequences holding together in the back of our eyes millions of times a second.
It's staggering. So here's our final recap for you. Single amino acid typos don't just change colors. They can cause structural collapses that lead to severe visual diseases or shift the actual color spectrum you can perceive.
It is a marvel of biology, and if we connect this to the much larger evolutionary picture, there is a tiny, fascinating detail mentioned early in the paper's introduction.
Oh, I loved this part.
If you look at ancestral vertebrates, think ancient fish, reptiles, and birds, they actually had 5 distinct classes of visual opsins. They possessed incredible, highly complex color vision that just dwarfs our own.
They could see colors we can't even conceptualize.
Exactly. But early placental mammals, our deep ancestors who lived during the time of the dinosaurs, they spent tens of millions of years scurrying around in the pitch dark as nocturnal creatures trying not to get eaten.
And because they didn't need day vision, we lost the hardware.
We did. Over millions of years in the dark, we lost 2 entire classes of those cone opsins. We were reduced to just basic dichromatic vision, essentially what we would call red-green colorblind today.
That's wild.
We humans and a few other primates are actually evolutionary mutants. We only have our red and green vision today because a random genetic duplication glitch happened millions of years later, allowing us to slowly rebuild a makeshift color vision system from the scraps that were left over.
We are walking around with a rebuilt patchwork system glued together by evolution. Which, brings me right back to where we started. I asked you to look at something colorful at the beginning of this deep dive. If it's still there, look at it again. Think about the millions of microscopic protein barrels in your eye right now. Think about the retinal traps firing, the structural pillars swinging open, the molecular babysitters balancing charges in milliseconds, all working perfectly together just to paint that specific hue in your mind. And I want to leave you with a final thought to ponder today. If swapping just one microscopic atom can fundamentally change the colors you're capable of seeing, and knowing that we lost massive chunks of our ancestral hardware millions of years ago in the dark, how much of the real, vibrant, wildly colorful universe are you completely blind to right now, simply because you are missing the tiny atomic structures that our ancient ancestors once had?